Good morning. I thought I would start off by introducing myself. My name is Jim Brody. I'm in the Department of Biomedical Engineering at UC Irvine. I've been there since I left Steve's lab in 2000. Let's see, I was a postdoc with Steve in Steve's lab at Caltech from 1998 to 2000. And really, there's two things I took away from there. One was this appreciation of trying to do science on trying to attack big problems using quantitative methods. And that's something I've carried with me since then. But the second is I met my wife there. And so some of you may know her. Here's a photo from our wedding. We had our wedding, you can see September 8th, 2001, right there on the Caltech campus at the Athenaeum. That is me. That's Anne. And in case you don't recognize him, there, right there is Steve Quake in 2001. So a quick update on what's happened to Anne. This was her back then, Anne Fu. That's how you might remember her. And a few of the papers she published there. She was kind of a joint student with Steve and Francis Arnold. And since then, she went to law school. She's now an attorney at Gibson Dunn, where she handles patent litigation. Here's a recent article she published. It came out just last week. So that's the kind of things she's writing these days. So here's what I want to talk about. I call it a different approach to cancer, and you'll see why. But it's really a quantitative approach that I've taken. A short summary of what I'm going to tell you is this: is that we should now be able to give a baby, someone just once we have their germline DNA determined, a test. From that test, we should be able to predict what type of cancers, if any, that this person will develop throughout their life. And why do I believe that? Well, the whole story starts with this, and this is a graph that I spotted actually right around about 2000. I was very interested in learning about cancer because of some of the projects I started working with Steve. This was a graph in Molecular Biology of Cell, the popular textbook. It was in the editions then. I noticed it's since disappeared in later editions. But it originates from about the mid 90s in a review article, and then it was thinking that had developed during the 80s. And the basic idea is that, as you can see on a log graph, they're trying to argue that the incidence of cancers increases with age and in fact increases exponentially. Now this, I would say, was very influential and really has permeated the thinking about cancer for a generation of scientists. I first got interested in this because of this prostate land. I was like, why is this one different than all the rest? So I went looking for the data and found something completely different than what this graph is showing. And so this is my version of the graph, and it looks like this. So this is actual data. And the point of this graph, I've kind of normalized it. Some of these, like colon, occurs much more frequently than bone sarcomas. But my point is just to look at the relationship between age and the specific incidence. And what you can see is that cancers occur at certain ages. I think this is a much more accurate description of the data. And they all, you can see, start decreasing until we run out of data. For the most part, a lot of the data cuts off at 85. One thing I spent a significant amount of time on was trying to figure out the mathematical shape of this data. Why is it in such a shape? And like I said, most of this data cuts off at 85. That's what I've got here. And if you take all the data points and try to fit it and with a reasonable idea of what the curve should be, there's kind of like one unknown. And the unknown is what fraction of the population will eventually get cancer. And the way I've plotted here is this 0 to 85 data for colon cancer, where the blue line goes, and this means everyone will eventually get cancer, 100%. And this red line is kind of a better fit to what it is, and I think it ends up being like 12% of the population would get cancer. And so what do I mean by that? Well, I mean, if you don't die from heart disease in a car crash or anything, and if you could just live on and on to 200 or 300, or, would you get this cancer? And that's the fraction there. So if I unveil the data after 85, it looks like this. And you can see that it's clearly not 100%. From this and other things, I've convinced myself that cancer only occurs in a limited group of people. We just don't know how to identify that group. And that this is really my prediction. You should be able to determine who will and who will not develop a specific cancer by simply analyzing their germline DNA. Well, people look for this, and they do it through these studies called Genome-Wide association studies. So let me describe this real quick. You get two groups of people with some trait, we'll say breast cancer, people without that trait, and then you genotype them using SNPs and look for differences between the two groups. This is an example. This is for ovarian cancer. Along here, I think they've probably got about 1 million different SNPs, and they look for the difference between these groups, and then they rate some kind of p-value. This is 10 to the minus 8 p-value. You can see there's a few here. So this is kind of considered borderline. I've seen ones that go into like 10 to the minus 300, 10 to the minus 400, where it's, these are the clear things that everyone knows about. But, so people have done this, and they've done this for all sorts of different cancers, and they don't find anything. Why is that? I'm claiming there should be something there, and they don't find it. Well, I have an idea why is that. Here's a quick description of how these association studies work. So like I said, there's two groups. There's the patients with some form of cancer, the patients without cancer. You genotype each patient. In this case, I'm kind of indicating a genotype measurement, like a SNP at 7 different locations, where the two possibilities are the capital letter, like the capital A and the small a. And then we just look for differences between the two groups, right? And so if you do this on this one, here's what you would see. Oh, look, at the E location, 100% differences. Patients with this variant of small e get the cancer. Patients with the large e don't, right? It's simple if it just occurs like that. But what about this? Epistatic interactions are multiple genes interacting. And so if you have combinations of genes, if you go through this, the same thing, the E's aren't like that anymore. There's nothing really there. And here's what you'd get if you look down here. There's, nothing stands out. But the way I set it up here is, what if you have something like this? Well, if you have this combination of variants or this combination, that identifies patients with cancer. And this or pattern doesn't appear ever over here. Now, this is invisible to the association study. If you look at some of the diagrams where they've worked out the interactions within a cell of how one thing interacts with another, they're very complex with plenty of nonlinearities, loops, feedbacks, redundancies. So I don't think it's at all a surprise that you might have complex things that cause genetic disease. So why can't we do that? Why don't we use machine learning, right? That's the big thing these days. Machine learning should be able to pull out these complex interactions. It can, it can tell the difference between a picture of a dog and a cat. Well, here's the problem. If we're doing this with any real data set, you can get maybe 10,000 nowadays, maybe even 50, 100,000, depending on the disease. But you're definitely looking this way at something like 1 million, hundreds of thousands, or 1 million, and you just can't do machine learning with that data set. You need more samples than features. So this is essentially what I've been working on for the past 5 years. How can we condense this information from millions of things down into a small, manageable number? So here's what the approach we've been taking. We started looking at chromosome length. This is if you take a plot of chromosome 1, take about 10,000 people and have some measure of chromosome length. There's a distribution. Some people have slightly longer, some people slightly shorter. Why is that? It's because of gene duplications, deletions, translocations, all these types of things that go on in the genome. How would you measure it? Well, the way we do it is this. And so, like I say, chromosome length is in quotes because it's a measure of chromosome length, but it's not really chromosome length. But anyways, a SNP microarray is really trying to just isolate whether one variant or a second variant exists in the sample. The way they do that ends up coming up with a number that is representative of how many copies of that sequence are in the sample. And they go through a lot of trouble to normalize this and everything, and they usually get reported in what they call L2R numbers, log base 2 ratio, then get processed. And so if you work with SNP data, you're probably used to just seeing this part, oh, it's a G, or oh, it's a C. But really behind that, there's some numbers. So we use those numbers to take it as a measure of the number of copies of this variant and really just sum them along the chromosome and take an average. And that's what I mean by chromosome length. So by doing this, we can build up a table, something like this. And it's just get a set of numbers like this, a label like this. And that's what you feed into a machine learning algorithm. It's just a classification algorithm. The question is, based on these numbers over here, can you classify the label here? Is it normal or is it breast cancer? So here we've got, I'm going to show you real quickly, 3 different tests, breast cancer that we did in 2 different data sets, and ovarian cancer and a brain cancer, GBM. This is just a quick summary of the differences between these 2 data sets. And I'll just go right to the results. In the TCGA data set, we could get this, an AUC of 0.75. If you're not familiar with the AUC, think of it as a true-false test. It's similar to that. And so if you get a 0.5 on a true-false test, 50%, that's a failing. I do these right. This is, to me, is like a C test. If you can get into the 0.9s, that's an A test. So this is a C test. Not horrible, not failing, but not great. Results from the UK Biobank. This was, I would say a B- test, 0.816. I've listed here all the different models. We use this package H2O, and it's in kind of an AutoML. It doesn't take much expertise to run. But my point I wanted to show here was that, this is kind of a list of the best models that this thing produced. The best one is a stacked ensemble. It's a combination of a bunch of different models, and it's really dominated by this deep learning model that kind of found a 0.79. And besides that, they're going into the 0.69. And my point here is that I think with some machine learning expertise, this could be improved. Other ways of improving the AUC is that we've been just kind of blindly taking chunks of the chromosomes. And it turns out that, let's see, I've got here, this is like chromosome 2. If you take the length, as we call it, of chromosome 2 for 10,000 different people and plot it against the length of chromosome 3 for 10,000 different people, they're highly correlated. So, these lengths aren't always independent from chromosome to chromosome. We can find, this is like sections of chromosome 1 are independent of one another. Sections of chromosome 1 and 2 are independent of one another, even though chromosome 1 and chromosome 2 itself are pretty well correlated. If you just look at the first quarter of those, they're not so correlated. The point is that there's a path to identifying subsets of the chromosome that are independent, which should probably lead to improvements in the AUC. Here is, we did this for 32 different types of cancers in the TCGA data set. And so here's the grading thing, AUC. And of course, I'm just going to pull out some of the better ones and talk about them. And the 2 really good ones here are ovarian cancer and glioblastoma multiforme. The exciting part of these is right down here. When this blue curve goes straight up, that means it's essentially perfect, no false positives. So the people who score really high on this test are almost certainly to get ovarian cancer. Okay, this was a test with about, you can see 425 women with ovarian cancer and about 10 times as many without. And then we score them all and rank them from highest to lowest. And if you look here in the top 1%, 44 of those top 1% had ovarian cancer and 3 did not when we ranked them. So you can look at the relative risks or the relative risk versus the bottom 50%, depending on how people do that. In conclusion, we should be able to do this. We should be able to give a baby a DNA test and tell that baby what cancers they're going to, if any, they will develop if they live long enough. We know how to do it. It's just a question of how good is the test, AUC, and there's plenty of paths to improve that AUC. Thank you.